Welcome everybody. Welcome everybody to this exciting and intense session of Chasing COVID. My name is Jordi Naval. I am the CEO of Biocat, and I will be your host this afternoon in this interesting and exciting session. This virtual workshop is made possible thanks to in collaboration with the Directorate General of Research and Innovation of the Department of Health of the Generalitat de Catalonia, also Directorate General for Research of the Department of Business and Knowledge, and La Caixa Foundation and EIT Health. So during this event, we will have 4 blocks of 5-minute presentations. It's going to be 5 presentations on each of the 4 blocks. And our aim is to have an overview of the main and most advanced research projects from our whole ecosystem related with COVID research. We will have a first panel about reprofiling, a panel about vaccines, a panel about new epidemiological studies, and a panel, I said everything. I mean, repurposing, new therapies, vaccines, and genetic studies. So the goal is that, and to make things a little bit more interesting, we have decided to vote for the best projects. We have 20 projects today, and we have decided to vote, for you, for the audience, to vote for the best projects in 2 categories. The first category is beautiful science. So I invite you, all the attendees today, to vote for the project which, in your opinion, is more attractive in scientific terms. You recognize beautiful science when you see it, right? So this is the first category. The second category of the voting will be a game changer. Those projects that really can have a practical application and can be deployed in the following months to help us cope with the COVID pandemic. So these 2 categories, the winner out of the 20 projects, the winner in each category will receive a prize of 1,000 euros. It's not that much, but I think it's a recognition of all the effort that you have been deploying over the recent months. And also, very importantly, this voting, bear with me. It seems complicated. It's not that complicated. Just it's quite simple. 4 blocks, 5 projects, each block, 5 minutes presentation per project. At the end of each block, we will have the voting of the 5 presentations we just saw, okay? And this, we will repeat that over the 4 blocks during the session, right? And so we will have the winner at the end. To make things a little bit still more interesting for you, and because today we have more than 300 registered people, registered scientists in this seminar, in this open poster session, to make things a little bit still more interesting, we would also vote in parallel, starting now. We will also vote in parallel which is going to be the first vaccine approved by the European Medicines Agency. It's going to be Oxford. It's going to be the Oxford team with this chimp adenovirus vector. It's going to be CanSino team with the adeno5. It's going to be Johnson & Johnson with adeno26. It's going to be Moderna or BioNTech with their RNA vaccines is going to be maybe CanSino. I mentioned CanSino. It's going to be the Sinovac Sinopharm. I mean, the Chinese company with a classical inactivated virus. So you see on your screen the voting. I see already the Oxford team is winning the race. So let's see what happens. This is going to be amazing to see the results because you are the experts. So it's very important. I think it's very important for everybody that we have an opinion. I know you have your favorites. I have mine too. So at the end of the presentation, at the end, like around 5:30, more or less, we will have the final voting, and we will have Dr. José Luis Cabero from ILX Therapeutics commenting on the final result. It's like after the Barça match, you have a post-partit. So we'll have the post-partit and we will comment on the final results. So all the panelists can vote too. Everybody can vote. So please, let's get actually started. Close all your other Zooms, Teams, phone calls. This is, I hope it's going to be a very exciting afternoon. And thanks for all the panelists. Thanks everybody for sharing your projects with you. So let's get started. The first panel is going to be about repurposing. It's about using drugs that have been approved for other indications, the use of these drugs specifically in dealing with a COVID infection or with the COVID secondary effects. The chair of this panel will be Dr. Ana Massagué. Ana, hello, how are you? I mean, from the Directorate General of Research and Innovation of the Department of Health. So, Ana, the floor is yours.
Thank you, Jordi. Thank you for inviting me to share this session. I mean, one of you.
No, come close to the microphone.
Okay. Now?
Better.
Okay. So thanks, Jordi, for inviting me to share this session, which, as you said, is about repurposing of drugs, animal models, where to. The speakers from this session are María José Buzón and Alicia Genescan from the group of infectious diseases of the BioRegion Institute of Research, Baldomero Oliva from Structural Bioinformatics Group of the Fabra University, Josep Per and Andrés Anton from the Liver Unit and the Respiratory Viruses Unit of the BioRegion Institute of Research, Joaquim Segalés from Cresa Atirta, and Alejandro Peralta from GPCR Drug Discovery Group of the INIM, led by Jana Segalés. As Jordi said, you have 5 minutes each to go with your talk. At the end, we'll have 5–10 minutes of discussion. And something that I was told, but I don't know, Jordi, you didn't say that, but according to the instructions, you need to vote for the beautiful science or the game changer during discussion. Because after discussion, the section is closed and we're going to vote anymore. Okay? So please, María José, if you are so kind, you can start. Thanks.
Thank you, Ana, for the introduction. Do you hear me? Yes? Okay. I need to see my slides. Okay. So this work has been performed primarily in the BioRegion Research Institute, and this is the team members and the collaborators. And the main goal was to establish a physiological model for the study of viral entry and also the testing of antiviral drugs against SARS-CoV-2. And this is because current methods are using cell lines mainly, but they have intrinsic limitations, as for example, the differential expression of host proteins that are involved in the viral entry. So the first thing that we did, because based on that, we want to establish a new model, but in this case, using human explant tissues using lungs. So the first thing that we did was to generate pseudoviral particles because it allows us to work in a VR2 facility. So we obtained the expression of the spike protein from the SARS-CoV-2, and we use the backbone of the BSV-G virus to construct our pseudotypes. And those pseudotypes, they express the luciferase gene or the GFP. And then, so we can see in the right graph that the titer of the virus are pretty high and pretty good. So in the next graph, this is an infection using Vero cell lines that are cells that are very susceptible to get infected with this virus. So the infection that can be blocked very nicely using an anti-AC2 antibody, because the AC2 is the main receptor of the virus for viral entry. And in the next graphs, this is an example of antiviral testing of different drugs using the cell line. So for example, we have some antivirals as the one shown in green, that they are potential, they have effect on the cell line. Some others in orange that they have some effect, but at very high concentrations. In here, we are showing 100 micromolar. And even we are able to see some drugs that are increasing the viral entry within the cells. So next slide, please. But as I said before, we wanted to establish a more physiological model. So we took human lung resections from surgeries, and then we disaggregate the cells and we perform the ex vivo infection assays. And in the next graph, you can see here an example of a flow cytometry plot of the cells. In here, you can see that there is a expression of the AC2 receptors in lung cells, and also those lung cells are able to get infected using BSVG GFP in this case. So we see around like 24% of infection. Also using this model and adding the antibody anti-AC2, we can block the infection in lung cells in a dose-dependent manner. But in the right graph, you can see different examples of different patients, and this blocking effect is somehow different depending on the donor that we are using. So in the next slide, please. So here is the parallel testing of some antivirals using the both models, the Vero cells and our lung tissue models. And these are some examples that we have testing. And for example, the first drug is Camostat. So Camostat, as you might know, is an inhibitor of one protease, a cellular protease that is required for viral infection, but this cell line does not express the protein. So no antiviral effect is observed. However, in our lung tissue model, we see a very nice antiviral effect. The second drug is a drug that is able actually to work pretty good in both models. However, if you look at the IC50, is somehow a bit higher, like 1 log higher in lungs compared to the cell model. And the third drug is, the third antiviral drug also it doesn't work in cell lines, but it's working somehow in our lung cells. And the last example is a drug which is enhancing like threefold the infection in beta cells, but no effect at all and not increasing the infection when using real cells from human lungs. So the next, so cell lines do not always mimic antiviral testing in our tissue lung explant model. This model is relevant and more physiological for the screening of potential antivirals and to study SARS-CoV-2 entry mechanisms. What we are doing now, so we are continuing testing potential antivirals. We have close to 40 right now. We are also working on the identification of alternative cellular targets for SARS-CoV-2 entry in the cells, in lungs. And also we are working on evaluating the influence of the immune microenvironment on viral entry and also the efficacy of antivirals. So thank you so much for your attention.
Can you hear me? Yeah?
Yes.
Okay. Thank you very much, Anna, for the introduction. This is a work published recently in PLOS ONE. It's an in silico drug repurposing combination of several drugs. And you can see that there are several institutions and groups that have been involved, public and private, for this work. So in the next slide, I'm going to introduce the methods that we have used. On the one side, we have program developed by Anaxomics. It's called TPMS. Is based on neural networks, artificial intelligence approach, and neural networks use a network of connections, of nodes, and then with an input and an output. In the input, there is the information that comes to the system, and in the output is for the training of it. Then the other method that we are using is Gildify. Gildify in the right side, it's a method developed in our group. This is based on PageRank algorithm, the algorithm that has been used by Google for when you are searching for web services. And it uses a message passing. It transfers information from one node to the other, and it covers the whole network. For the network, we use the protein-protein interaction network in both cases. In the next slide, please, we can see what are the characteristics that are common for these both methods. And this is the use of an input, that is our gene disease associations or drug target associations. Then all the nodes get some kind of a scoring from the system, and that is going to tell how relevant are these nodes or proteins for the pathology that we are going to study. And on this, we can get some kind of pathways with the relevant nodes that connect from the input to the output, the result. There is a difference between both methods. TPMS uses a result, an objective defined by the nodes that is going to receive the answer from the input, while Gildify doesn't have this output. This means that the system transfers all the information along the network, and then we have to use a threshold to get just the top ranking ones. Then we can use these both approaches to study the potential application of drug combinations. So we have a set of nodes associated with a disease in yellow that makes a network, and then we can have one or two or several drugs, and what is the influence of these drugs through the network, transferring the information to the pathology. So that's what we have used for SARS-CoV-2 infection. And in this particular case, what we have is the network is just the interactome network, all the protein-protein interactions, then the input. For the input, we have all those targets that we know have been involved in the infection, such as ACE2 or TMPRSS2 or furin. These are for the entry. And then in the output, we have the relationship between the human interactome and the virus interactome. That's something that was recently obtained by the group of Krogan. Or we can have other kind of pathologies, such as the acute respiratory distress, which is an outcome that gives you the information on how it is affecting the virus. So in the next slide, I will show the results that we have obtained. So first, with the use of TPMS, we have been using all potential pairs of drugs that are underused commercially and are stored in drug bank. And then as the output, we use the virus host network. So we are trying to obtain the highest impact on this network. And then we rank all these pairs of drugs, and we focus on those drugs that were involving affecting furin, is one of the targets in the entry. This happens to be pirfenidone, and then the highest score is gotten for the combination with melatonin. Then on the right side, we have what we did with Gildify. So now we know the input, so we put both targets as an input, and we obtain the connection of the whole networks of both drugs, from where we can get what are the relevant pathways or the regimen of functions. And also using TPMS, we can see what are the mechanisms of action more relevant for the connection with acute respiratory distress. So among them, we can obtain using Gildify, and then this study with TPMS. Some relevant properties that comes out from this study are the characteristics of these two drugs that are anti-inflammatory, antioxidant, antifibrotic. So we think that this can be used just to relieve the severe pulmonary complications coming from COVID. And with this, I'm done with the presentation, and later for the questions, we will also have Mireia Coma from Anaxomics that can help on the answers. Thank you.
Okay, much. Although I hope you is listening me well because I have troubles with microphones. Now it's done from just and doesn't done for you viruses BioRegion.
Hello, good afternoon. Can you hear me well?
Yeah.
Okay, so to achieve the 4 objectives of our project that has been funded by the degrees, we have followed 2 main strategies. One has been to sequencing the whole genome that now is working properly well in the laboratory of Dr. Andres Anton. And also we have followed a deep sequencing analysis of some genes of the SARS-CoV-2. Next slide, please. And specifically, we have focused our attention in the sequencing of the spike gene that codifies for the spike protein. The spike protein is composed by 2 regions. The region 1, the S1, includes the receptor binding domain, and the S2 is the one that fixes the spike to the surface of the virus. To sequence the whole spike, we have designed 13 primer pairs. Each primer pairs, we call it an amplicon. So next step, next please. And we have studied the spike of 18 patients. And the result that we obtain is something like this graphic that we make a zoom like this. In this graphic, we show, we have studied the deletions. Each column, the blue column, represents a deletion. The width is the number of nucleotides that are lost, that are deleted. And the height is the number of reads of sequences that we obtain. In the right side of the graphic, we represent the percentage. Next, we have studied more than 200,000 complete spikes per patient. And the results that we have obtained are represented in these boxes, in the 13 boxes that represents the 13 amplicons. Inside of each box, we represent the deletions in each of the patients. And I want you to highlight the amplicon number 7. Here, as you can see, there is an accumulation of deletions in most of the patients. And all these deletions are observed exactly at the same positions, which is upstream, but very, very close to the S1 and S2 cleavage site. Next, please. So this has led us to propose a hypothesis. In the left side of this graphic, you can see the normal viral cycle of the virus that finishes with the production of a genome of an infectious virus that is secreted by exocytosis. And in the right side, you can see that when there is a deletion, these deletions causes the appearance of a stop codon, and then this generates the translation, the production of a truncated spike protein that is composed by the subunit S1. And as this S1 is the external part of the spike protein, it is probably that this peptide is soluble, and we propose that this can be secreted and it can do the best that it can do. So it's binding to the ACE2 receptor. Next, please. So this work, next slide, please. This work has been recently published in Emerging Microbes and Infection. It's the work of different groups on Vall of Campus Vall. And the highlights and implications of our results is that natural deletions are a source of viral variation, that there are some hotspot deletions upstream and very close to the S1 and S2 cleavage site that these generate truncated S proteins. And we suggest that these proteins, this is a kind of a strategy that we have called the don't burn down the house, in which the free S1 protein may compete with the viral particles for the ACE2 receptor, and thereby attenuating the tissue damage, and this helps the virus to produce viruses and be transmitted more easily. In addition, it is possible, or a possible consequence is that this free S1 protein may bind to host neutralizing antibodies, and this has helped us to start a new line of research trying to find these free proteins in infected patients. And thank you very much for your attention.
Thanks very much, Josep. And now is the turn from Joaquim Sagales, please.
Thank you very much, Anna, for the presentation. Good afternoon to everybody. Well, there's always an animal side of everything. So from this point of view, this part, it will include the part which is much more related with the One Health concept, because at the very end, at the start of these pandemics, it was a lot of unknowns, and some of the unknowns were related with the potential zoonotic origin, but another one was which kind of animals were susceptible to SARS-CoV-2. At the very end, it was very important to know if there are several potential species which are susceptible, and especially those that are able to recapitulate the problems that the human beings are suffering from COVID-19. So by February already, March, the World Health Organization established a COVID-19 animal model working group. And very recently, this working group has published a paper, which you can see on your right-hand side, regarding animal models for COVID-19. So in our case, instead of presenting rather than a project, it's much more a platform related with the animal model development, specifically oriented to work with SARS-CoV-2 and COVID-19. Next slide, please. In fact, the major objectives of this platform was to develop such different animal models with the specific objective to study pathogenesis and immunopathogenesis against the virus, and very importantly nowadays, and especially within this section of repurposing drugs, on the application of such animal models to test antiviral drugs, but also antibodies, and very importantly, vaccines. In fact, the most, let's say, precise objectives would include the development of animal models able to recapitulate what's going on in the human beings. This is the difficult part, taking into account that the human beings, they have a very broad spectrum of clinical signs as well as lesions, and also to try to find animal models usable for immunogenicity studies when developing vaccines. Next slide. So far, and since March, we have been working on the biosafety level 3 laboratory and animal facilities at IRTA-CRESA. Those are very special kind of facilities that allows us working with SARS-CoV-2 and do infections, not just in vitro, but also in vivo with this kind of virus. In fact, we have been working for more than 6 years already with MERS coronavirus, another important zoonotic deadly coronavirus. Well, so far and since then, we have established the hamster model with the golden Syrian hamster. This is a kind of animal which is naturally susceptible to SARS-CoV-2. In fact, the model is relatively simple because it consists on the international installation of a certain amount of virus which could vary between 10 to the 4 to 10 to the 6 TCID50 per animal. And the readout of the system is excellent because it includes 3 major legs, which is the clinical one because of losses weight, the pathological one because they develop moderate levels of pneumonia, interstitial pneumonia, and finally from a virological side because the viral load, especially in the respiratory tract, namely upper respiratory tract, nasal turbinates, and lungs, is a very important readouts. Moreover, we have been able to establish a kind of system of infection and reinfection with the hamster model, and we observed that certainly the animals can protect against the lower respiratory tract open reinfection, but not the upper respiratory tract. We also set already the humanized AC2 mice model with a dose of 10 to the 3, 10 to the 4 TCID50 per animal, again for international installation. And we also recapitulate several different issues which could be certainly comparable only to a certain degree with humans. There's a loss of weight, so there's a clinical outcome. However, there are very significant neurological signs, which so far it's not the typical presentation in humans, as well as pneumonia and huge viral load in respiratory tract as well as brain. And finally, we set a swine model we realized already that pigs are not susceptible to SARS-CoV-2 infection. However, when we did experimental infections by means of parenteral inoculation, namely intravenously and intramuscularly, those animals were able to seroconvert. So pig probably can recapitulate to a certain degree a potential immunogenicity model, and take into account that pig could be a physiological and immunological good model for human, so we have to take into account that could be of interest. Next slide, please. So which is the research impact that we expect from those animal models? Of course, the possibility of do reinfection studies, or even to do multiple passages, or even the possibility of studying aerosol transmission, which is quite hotspot nowadays. Of course, we can model SARS-CoV-2 transmission in different settings, and at the very end is the possibility of recapitulating as much as possible the COVID-19 forms. So far, we have been trying, especially with the hamster model and the mice model, young animals. What could happen if we are using older animals or animals suffering from comorbidities, for example? And very importantly, I would say, and this is the short-term most important issues at present, is the use of those models as preclinical study technology. Because at the very end, if we have to test vaccines, antibodies, or antiviral drugs, it will be very, very important to use such models in all cases. And with that, I would like to finish just to say that this kind of platform has been obtained at the very end with the collaboration with Directorate together with Barcelona Supercomputing Center and IRSI Caixa in a kind of big consortium that has been also participated by Grifols. So with that, I thank you very much for your attention. Thank you.
Thanks, Joaquin.
Thank you. Hello, everyone. And today I will be presenting the project of a dual strategy for inhibiting the infectious cycle of SARS-CoV-2, which is supervised by Jana Selend and in collaboration with Thomas Espetifnis and Maria Vadhua from Interax. As the name suggests, our project takes a dual approach for targeting and inhibiting the entrance of the virus into the cell using already approved drugs and candidates in advanced investigational stages. As you know, SARS-CoV-2 viruses uses its surface protein called the spike protein to bind to the human receptor ACE2. Subsequently, the human serine protease TMPRSS2 facilitates the virus entrance into the cell. So, our first strategy consists in blocking a specific region of the spike protein, called the receptor binding domain, that is used to attach to the host cell. In order to find compounds that could inhibit this attachment, we performed a virtual screening consisting in protein ligand docking into the interface between both proteins, the spike proteins and the cell ACE2. Our second strategy consists in inhibiting the serine protease TMPRSS2, which catalyzes the entrance of the virus into the cell. For this target, we could not use the docking approach given that it does not exist a crystallized structure for this protease. So we decided to perform a virtual screening using a ligand-based pharmacophore approach based on ligands that are known to inhibit this protease. This pharmacophore approach consists in detecting common structural features that are related to the protease inhibition and that can be used in a next step to screen for novel compounds that match also these patterns. In the remaining slides, I will explain the main steps of the project. The first step was to perform a virtual screening with the methods already explained, the docking and the pharmacophore-based approach. We screen against a database consisting of the merge of DrugBank, PubChem, and also other metabolites found in SIG15 database. The second step, after we found some candidates, was to perform molecular dynamics binding simulations of the top drugs against their targets. Then, the third step was to look for antiviral activity against SARS-CoV-2 in the bibliography. In this step, we found that 3 of our top hits were antivirally active at safe concentrations, as Riva et al. Stated in the recently published Nature paper. The candidates that emerged from our screen are highlighted here in red. Apelimod and astemizole performed excellently in our docking results, which we can see in the next figure, scoring higher than 97% of the molecules in the docking. In addition, we found flumatinib with an even higher score than that. Finally, in our fourth step, our colleagues of Interax, in the next slide, have established an assay to measure viral infection using SARS-CoV-2 pseudovirus. This pseudovirus carry luciferase RNA that, when the infected cell, is expressed inside them. This way, we can quantify how many viruses enter the cells, and this way test whether our top compounds inhibit the enzyme's mechanism or not. So using this assay, we obtained our first evidence that apilimod inhibits the entry step of the infection. In the following, you see a quantification of virus entry measured as luciferase activity showing on the y-axis. The more luminescence means the more virus have entered the cell. On the left, you see a control experiment with an ACE2 antibody. The higher the antibody concentration, the less virus enters the cell. Meanwhile, on the right, you see our results for apilimod, which reduces virus entry by approximately 80%, which we think is very promising. So currently, we are now in this phase of confirming these results and also testing other several promising candidates. That's it for now. Thanks you for listening.
Very much.
Okay, so thank you very much. We had planned that Ana Meseguer will host a short debate, but I'm afraid that we have sound problems. So we'll just thank you, Maria, thank you, Baldo, Josep, Kim, and Alejandro for your very interesting presentations. I think that we should, now it's time for the audience to vote for the beautiful science and for the game changer project among these 5 projects that we have seen now. So if you can, the guys who are managing the Zoom in Girona can really put the screen with the voting and close the presentation and plug in the screen with the poll. Also, I would like to remember the people, the audience, that there is a chat screen where you can also send questions to the panelists. It's not just us who are going to ask the questions or open a little bit of a debate, but you, the audience, very much invited to pose your questions to the panelists. So let me start with Maria Jose. You've mentioned that anti-ACI-AC2 antibody does not block viral entry in all patients equally. Does it mean that there are other receptors playing a role in viral entry? What's your take on that?
So I don't know the answer yet, but we are working on that because it was a very interesting finding. It might be that there are some other receptors that are mediating viral entry, or maybe some other isoforms of the ACE2 receptors that are expressed in different cells. So I don't know the answer, but we are working on that.
Okay, thank you. Of course, it's one of the key questions that everybody is wondering about these days. Next, Baldo, do you think, or is it possible that melatonin alone might have an effect on the evolution of the infection, or it has to be a combination, and why a combination?
We presented this as a combination, but actually this is an interesting question that it was posed by an email by an Indian clinician. And I know melatonin. It's appeared also in the work of Krogan as one possible drug to be used. And I think Mireia, I don't know if she can be here too, but in Anaxomics they also studied more deeply on the use of each one of the individual drugs. Perhaps she can comment on that if she is in, but I'm not sure.
No, I don't think we can have Mireia here.
Okay,
so it's so complicated. You cannot imagine how complicated it is on the backstage to organize this meeting. I'm sorry.
So.
I think we are doing we're doing quite well, but it's difficult to plug in people from so many different places. So thank you. I mean, please go ahead, Aldo. Go ahead.
Yeah. No, I know they did study each individual case, so that's why I know they have more deeply analyzed each one of them separately. But the work was done on the combination. It appeared melatonin and pirfenidone, but there were also many other pairs that came out of this work.
Okay, excellent. So next question. I mean, I have, of course, one question for each one of us. And please, the audience can also pose your questions. Or, of course, this is one of the goals of this webinar is that everybody gets to know everybody. Maybe you have never worked together. You don't even knew that Josep existed from Baldo's perspective. I don't know, maybe yes, maybe not, but it's important that to have this sense of community. We are all together from very different places, universities and hospitals. We are all together today here trying to solve a common nightmare for all of us. So it's also good to get to know each other. So please, the panelists can pose also questions to the other panelists. Feel free to do that. Josep, you've mentioned the strategy that don't burn down the house is something that the virus has acquired during the pandemics. I mean, what do you mean by that?
Yeah, well, the answer is that we don't think that the virus has acquired it during the pandemics because the samples that we have studied came from the beginning of the pandemics. So they were taken in March 2020, so when it started here. So that we think is that the virus was carrying this strategy from the beginning. And is it possible that this kind of strategy can be selected in different during the evolution of the virus, jumping from animals to humans or whatever? Because it's something that can be quite general, not specific of the humans in this case.
Okay, thank you. Yes. Joaquin, Kim, I mean, do you think that other animal models can be of interest? I mean, besides rodents? I don't know.
Yeah,
monkeys, mini pigs. I mean, what other ideas or what's out there in this field?
Well, at the very end, a number of animal species have been tested for susceptibility to SARS-CoV-2. However, just a minimal number are naturally susceptible. And among them, for example, besides the golden Syrian hamsters, we have the ferret as well as the mink. So those are animals that they can recapitulate to a certain degree, a certain disease. However, it's extremely mild and sometimes no clinical signs. Like the cat, the domestic cat and other felids are susceptible to the infection. However, they do not display almost clinical signs or minimal ones. The other ones, which of course are quite far away from our possibilities, or for most of us, is the non-human primates. Non-human primates, at least some of them, recapitulate rather nicely as well the disease in humans, a kind of moderate disease. However, if the specific question would be, can we get an animal species recapitulating a really severe form of COVID-19? Nowadays, we do not have it. This is something that might be in the future will come, but might be we will need also animal species suffering from might be comorbidities, co-infections, or even elder animals, which is not easy to work with.
Okay, thank you. Thank you very much. Very interesting, your perspective on that. I will pose one last question to Alejandro, and we have one question from the audience, and we will move to the next block because I mean it's going to be an intense afternoon. We have many presentations. So this question is for you, Alejandro, or somebody from your team that can really chip in. You've mentioned two strategies to inhibit the infection of SARS-CoV-2, blocking the spike protein or inhibiting the serine protease. Which is your preferred strategy? What are the advantages or disadvantages of each one of them?
I think that maybe the first strategy, as we are focusing on virus protein, has less side effects because we are not inhibiting natural processes in human cells. So in that way, I think that the first strategy will have less side effects, so will be more useful. And also, as it is a structure-based approach, it is specific for the spike protein. Probably will have more direct effects than doing a ligand-based approach that is maybe more general for a wide range of targets. So strategy one, I think, would be the most promising one.
Okay, thank you very much.
Thank you.
I think if it's possible, can we insert the poll, the voting screen, so everybody can start voting very quickly? The preferred beautiful science and game changer project. Well, whilst we wait for that, there is one last question that would like to pose. I think it's for Maria. I mean, there are many questions from the audience. We don't have time to ask all of them to the panelists, so we'll just go for one of the many of the several questions. Is as follows. I mean, is the idea of finding a drug? This is from Giulio Rosati. Is the idea of finding a drug blocking ACE2 receptor involving people solved? I mean, are there drugs in the clinical trials blocking ACE2 receptor under study, or is this is just an idea that we have discarded for the time being?
That's a question for me?
Yes.
No. Oh, yeah. To my knowledge, there is not. But I mean, the drugs that has been described in the literature, they are trying to block or the S protein or the interface between the S protein or the AC receptor. But I'm not 100% sure of the answer.
Okay. So I don't know if the audience is seeing the voting poll. Okay, so people can vote. Sorry, it's because I'm seeing a different screen. So I understand that people can vote now. So please remember to vote in two categories. I mean, the beautiful science. Which of these 5 projects did you like the most? Do you think were more innovative or creative? And which one of the projects is the one with more chances of being a game changer in the short term and really change our lives and the global course of the pandemic? Remember to scroll down the screen, because the voting, the screen is long with the different projects, so the voting button is at the end. Excellent. So I think everybody has been able to vote. Beautiful science, game changer. So thank you the panelists. Thank you, Maria, Baldo, Josep, Kim, Alejandro, for your beautiful and exciting presentations. I hope you're the winners. Please stay with us until the end. Also, please vote. Again, there is 2 votes ongoing at the same time, so we like to make things as complicated as possible. We enjoy complex things. So there is 2 votes, beautiful science, game changer, and at the same time, in parallel, we will be voting or betting, more than voting, betting, which is going to be the first vaccine to reach approval by the European Medicines Agency. Oxford team, I think, is on top. Moderna is following closely. Couple of Chinese projects behind. BioNTech, Novavax, or you can even vote others if you think that those are not those are not the front runners.